Hello everybody, this is Dr. Christopher White and welcome to this video looking at the lab exercise faunal succession and depositional sequences. So in previous lab exercises we have already looked at the methods which geologists use to date layers of rocks or sequences of rocks. Now obviously the two methods that are employed by geologists are relative dating and radiometric dating. So of course relative dating is going to try and put your layer of rock or your layers of rock into a position on the geologic timescale, but it's not going to assign a numerical age to your layer of rock or your sequence of rocks. Radiometric dating, on the other hand, is going to allow you to assign a numerical age to your rock, and therefore you can then use that to place its position on the geologic timescale. Now, as we've already touched on, relative dating is often the method of choice for geologists, and that's purely because radiometric dating is much more expensive versus relative dating, and radiometric dating primarily focuses on minerals which are more common in igneous and metamorphic rocks, which is a bit of an issue when most of the rocks which are outcropping on the Earth's surface are sedimentary. So very often radiometric dating of sedimentary rocks is highly complicated. It can be done, but it's not an easy process and is therefore it's even more expensive than other more commonly used radiometric dating methods like zircon, for instance. Now, in order to actually date our rock and work out where we fall on the geologic timescale using relative dating, one of the strongest tools is, of course, the principle of faunal succession. And the principle of faunal succession simply states that we know the order in which fossils occur. So if we find a fossil in our rock, we can look at previous studies and we can say, right, that fossil existed in the Cambrian. And as such, we know that the layer of rock in which that fossil has been found will also therefore date to the Cambrian. Now, for each fossil, this means we are going to have a date of first appearance and a date of last appearance. And this is referred to as the fossil's age range, or more commonly simply referred to as its range. Now, the first appearance and last appearance of the fossil is typically not assigned a numerical age. It typically only tells you, right, which period or periods did this fossil occur in. Now, there are certain rare circumstances where you do have fossils which have numerical ages applied to them. These are sometimes referred to as anchor ranges or anchor fossils. And the numerical ages for these fossils are known because either we have used one of these more complicated radiometric dating methods to actually date the sedimentary rock which contains them, so we get a numerical age for the fossil that way. Or the other way is if the layer of sedimentary rock containing the fossil is sandwiched between two layers of igneous rock. So they could be something like lava flows or maybe a pyroclastic rock, something like a tuff maybe. And of course, these layers of igneous rock are going to contain datable minerals. And so we can date the layer of igneous rock below, we can date the layer of igneous rock above, and that allows us to essentially produce an age range in which this fossil could have existed. Now, the problem that geologists have a lot of the time is that when we are looking at a sequence of rocks, we will typically see the same fossils occurring in multiple layers. And this is because a lot of fossils have quite large age ranges. And this can be a bit of an issue because if you have a fossil that's known to have occurred across multiple periods, so let's say our fossil of interest is known to have occurred in layers of rock from the Cambrian, Ordovician, Silurian, and Devonian, it's obviously going to have an age range between Cambrian and Devonian. So if you're in the field and you come across a layer of rock that contains the fossil in question, well, how are you going to know which period it came from? Did it come from the Cambrian, the Ordovician, the Silurian, or the Devonian? So in order to get around this problem, what geologists have started to do is they've started to use the fossil assemblage, which is displayed by a layer of rock. So in the case of the fossil assemblage, what we're doing is we're using the ranges from multiple fossils from the same layer, and we are looking to try and work out which period or periods all of these fossils could have coexisted in and therefore been incorporated into the same layer of rock. Now, this is a very helpful technique because it allows you to use fossils, some of them sometimes with quite large age ranges, to date your layer of rock, because once again, you are just trying to find that one period or periods during which all those fossils could have coexisted. So if we look over here on the right hand side, we can see we have a diagram which is for 6 hypothetical fossil species. And for each of these fossil species labeled A to F, you can see them across the top there, the age range for them has been coloured in green. So for instance, fossil A is known to have existed between the early Permian and the late Jurassic. So that's its age range there. And so what you can see we're doing is we are drawing each range onto the diagram, and then we are simply going to locate the period or periods in which each of these 6 fossils could have coexisted together. And so we can see quite clearly that the only period where all 6 fossils could have coexisted is the Jurassic. And as such, we know our layer of rock must therefore be from the Jurassic. Now, if we drop down here, we have a situation where, once again, we can see we have 6 hypothetical fossil species, and A to E are exactly the same fossil species that we can see up here in the first diagram. However, fossil F has been misidentified. So you can see the age range of fossil F is Cambrian to late Devonian. You'll notice straight away that this really doesn't work with the rest of the fossils that we have identified. So if we look over here, we can see the other 5 fossils are suggesting that the age of the layer must either be Triassic or Jurassic. And so when we see this kind of thing happening when we are analyzing a fossil assemblage, that instantly tells us that, yeah, we've probably misidentified fossil F. And as such, we need to go back to the sources and we need to try and re-identify it, and we need to see if there is another fossil which is similar in appearance, which has an age which would be consistent with the data from the other 5 fossils. So we're going to be using this process in this lab in order to date several layers of rock from the same sequence. And so by dating these layers of rock, what we're going to be doing is we're going to be trying to build up a bit of a geologic history for this sequence of rock. So we're going to try and date each layer, and then we're going to try and also analyze the type of contact between each layer, and the dating of each of these layers is going to allow us to do that. Okay, so for part 1 of this lab exercise, we are going to begin by actually dating our layers of rock. So I'm just going to drop down here to the diagram for our sequence, and I'm just going to bring it into the center there. Okay, so you can see we have a sequence of rocks. We can see we have a granite down here at the bottom underneath the sequence, and you can also see over here on the left we have an area of Precambrian rock which has been dated to the Archean, and an important point here, it is metamorphic. You can also see that the sequence of rocks itself consists of 8 formations labeled FM1 to FM8. These are all sedimentary layers. You will also notice that for each of these formations, there is 1 or 2 locations where fossil samples have been collected for dating purposes. So you can see that for formation 1, there is one sample location which has been used. In the case of formation 2, you can see that there are 2 locations which have been used to collect fossil samples. Now, this means when you are moving on to question 1 and you start dating each of these layers, in the case of formation 1, because there is only 1 location where fossil samples have been collected, you will have a single age for that formation. So, for instance, let's say that formation 1 dates to the Ordovician. Now, in the case of formation 2, on the other hand, as we have 2 different sample locations, these sample locations may not have the same age. And so this would mean that you would have an age range for formation 2. So let's say that location 2 is dated to the Silurian and location 3 is dated to the Devonian. And so this is then going to mean that formation 2 is going to have an age range between Silurian and Devonian. So in the case of formation 2 and formation 3, you are going to note an age range rather than a single period. Now, I would ask, once you have dated each of the layers, when you are writing the age for each of them, please just give the period name or names. Don't give me numerical values. Let's keep it simple. Say this layer of rock is from the Cambrian or the Ordovician, or it's from the Cambrian to the Ordovician. So that's the information that you're going to be putting in this table here for question 1. Now, I should point out that when you are dating the sample locations, you are going to be using an age range diagram like the one you have already seen. Now, some of you will already have spotted that for the Cenozoic, it lists the 3 Cenozoic periods. So we have the Paleogene, the Neogene, and the Quaternary. However, the fossil tables that you will be using to actually identify the fossils will give you fossil ages for Cenozoic aged fossils to an epoch level. So for instance, you might identify a fossil and it might tell you that that fossil dates to the Eocene. Now that's an epoch, not a period. And so if you find that fossil and it dates to the Eocene and you then end up dating that layer of rock, that formation, and the formation of rock dates to the Eocene, well, on the diagram, you are therefore going to fill out the Paleogene box. So if you have a formation that dates to the Paleocene, the Eocene, or the Oligocene, you are going to fill out the Paleogene box on your range diagram. If, however, you have fossils that date to the Miocene or Pliocene, you are going to fill out the Neogene aged box on the range diagram. And if you have a fossil that states that the age range is, let's say, Pliocene to recent, well, recent means that the fossil is still around even now. And so obviously this means that the fossil is around now, so it's in the Quaternary. I should point out that for Quaternary age fossils, there are no epoch level ages given. So it doesn't say something like Holocene, for instance. It simply says recent, and recent means that the fossil dates to the Quaternary. Okay, so how are we actually going to go about dating each of the locations? So for each of the locations, you can see that we have a fossil assemblage. So I'm just going to drop down here on the right hand side. And so this is our fossil assemblage for location 1. And you can see that it consists of 5 fossils, which are labeled A to E. And if you come down to the following page, you will see that you have an age range diagram. It's labeled location 1, and you will see that there is space for 6 fossils. So in this instance, we only have 5, so this final column F will not be used. So when you are looking at the fossil images, please know that I have done my best to make sure that the fossil image in front of you is representative of the image within the fossil tables. And so that's going to make the identification of the fossil just that little bit easier. So when we're looking at location 1, what are the features that we can spot for each of these fossils that's actually going to allow us to correctly identify them? So when we're analysing any fossil and trying to compare it to the diagrams in the fossil tables, what you're really looking for are the key features for each fossil. So if we look at fossil A, what can we see? Well, to begin with, you can probably spot straight away that it has an appearance which is somewhat similar to a woodlouse or a pillbug. So you can see it clearly has a segmented body. Now, in terms of the organism itself, I'm going to, for simplicity's sake, split it into 3 separate regions. You have the head, the middle, and the rear. Now, in the case of the head, what can you see? Well, straight away you can see you have this smooth curve. There's no obvious spikes or serrations or anything like that. You will notice that it does have 2 eyes. It has an eye right here and another eye right here, but they are, compared to the size of the head, actually quite small. You will also spot that it has this noticeable lump in the middle of the head, which for simplicity's sake I will refer to as the nose. And you can see that it has quite a large nose, and that nose is obviously quite noticeable, and it's located between the eyes, and it kind of sits in the middle of the head. It doesn't really seem to extend towards the edge. Now, when you move towards the middle, you can see that it consists of several segments. There's nothing particularly special about them. Once again, there are no obvious spines or anything like that. And then when you move into the rear, you can see that you have a relatively smooth area. So there's no segments in this particular portion of the fossil. And of course, this is going to be important because when you're looking at organisms like this and when you're looking in the fossil table, you will notice that some of them will have segmented middles and they will also have segmented rears. But in this particular case, you will notice that the rear is smooth, not segmented. And so when you go and look in the fossil table, you'll be looking for an organism that has a pill bug-like appearance. You will look for things like the small eyes, the noticeable and quite large nose, and you will also look for one that has a non-segmented smooth rear portion. And hopefully that would then allow you to identify your fossil correctly. Now, in the case of B, what are you looking for? Well, straight away you can see that the fossil itself has a donut-like appearance. You can see it's circular. It clearly has this core region in the middle, and then you can look at the portion of the fossil itself in between the edge and the middle of the fossil. So if we just zoom in, what can we see? Well, I'm afraid you'll have to forgive the pixelation, but you can see that the fossil itself has a radiating appearance, doesn't it? So you clearly have lines which are coming from the center and moving out towards the edge in a radiating pattern. You'll also notice that there do appear to be individual chambers. So it looks like what you have here is a fossil that is actually a slice through a larger feature. So if you imagine what you're doing is you are cutting through, let's say, a pencil. And so the pencil itself is obviously quite long and slender, but if you slice through it and look at it from the top down, what you will see is something that looks like this. It will be circular. You'll have a core region and then you'll have the wood of the pencil around it. So it's a similar appearance to that. So what we're actually looking at here is a slice through another fossil. And so the question is, well, what shape is that fossil? If you were to look at it in 3 dimensions, would it be kind of like a cylinder, like a pencil? Could it be something like a cone? Who knows? So what you're going to do when you look through the fossil table is you are going to look for a circular fossil that has a donut-like appearance. So you're looking for a core region that doesn't have anything there, essentially empty, and you're looking for it to have these radiating segments coming away from this core region towards the edge, and you're also looking for a fossil that has individual chambers within those core segments. Okay, so now if we move on to C, once again you can see straight away that we have an organism that has that pill bug-like appearance again. Now, just like A, you can see we have a distinct head region, we have a middle, and I've got to be honest, the rear portion of this fossil isn't particularly clear. And the reason for that is it's segmented, so it kind of blends into the middle portion of the fossil. So it's difficult to define exactly where the transition from the middle to the rear of this particular fossil is located. Now, straight away, you will have noticed that the head has this very broad semicircular appearance, and a large proportion of it is very smooth. You will notice that there was an eye here, there was an eye here, and there was a nose-like feature located here. However, you can also see that they have been damaged, so they've been lost. But the fact that you have this very broad, very smooth semicircular head is quite a good feature for this particular fossil. The other thing that you've probably spotted is that this particular organism has a lot of spines coming off both the middle and the rear portion of the fossil. So each of these segments appears to have its own individual spine, which is running along the edge, the margin of the body. And so if you go into the fossil tables and you look for an organism that looks kind of like a pill bug, has this broad, relatively smooth head, combined with a fully segmented middle and rear, which has multiple spines coming off, then you should be able to identify your fossil relatively quickly. Now, D is a slightly more complicated fossil. So when we're seeing an organism like D, it instantly brings us into is it a brachiopod or is it a bivalve? So when we're looking at fossils like this, the first thing we're going to do is we're going to look at the shape. Is there anything distinctive about the shape? And as you move through this lab exercise, you will come across bivalves and brachiopods that have very distinctive shapes to them, and that is going to help you identify the organism that you're looking at. Now, in this particular case, the shape itself isn't particularly different. It's quite a regular shape, to be honest. So you have the hinge region, which essentially is where the two halves of the shell would have met. And then obviously you have the edge where the two halves would have closed up to seal in the animal inside the shell. Now, in terms of the shell itself, is there anything obvious? Are there any distinct ridges, or are there distinct raised or lowered portions of the shell? And in this case, you can see, no, not really. It's actually a surprisingly smooth looking shell. Now, the one feature that it does have is you can see that it has these concentric bands running along the circumference of the shell there. So you can see them one there, you can see another one here, you can see several more as well. Now, that's a feature you're going to want to keep an eye out for when you are trying to identify this fossil. Because sometimes when you come across brachiopods and bivalves, what you are going to see is you are going to see that they can either have radiating striations, so you'll have a striation that starts in the hinge region here and moves towards the edge, and you'll have multiple examples of that, and it'll look kind of like a sunburst kind of pattern on the shell. And then you will have some shells that have these concentric bands, which runs parallel to the margin of the shell. And then you will have some brachiopods and bivalves where you have a combination of the two, where you have some striations, some lines running from the ridge, running from the hinge towards the margin in that kind of sunburst pattern. And you will also have these concentric bands as well. So that's what you really want to be keeping your eyes open for when you are looking at these brachiopod and bivalve shells. So begin by looking at the shape. Is there something very distinctive about the shape that could help you? In this case, not really. The next thing you're going to look for is, are there any obvious striations or bands on the shell? In this case, yes, you can see we clearly have these bands. Now, D is going to be a little bit difficult to identify. So when it comes to identifying D, what you should do is you should identify A, B, C, and E first. So once you've identified those four fossils and put their age ranges onto your location 1 range diagram, it's going to give you some kind of idea about the age of the layer of rock. So let's say, for argument's sake, it says the age of the layer of rock could be Ordovician or Silurian. Okay, so then when you analyze D, the question is does D have an age range that falls within the Ordovician or the Silurian? So if you analyze D and you identify the fossil which you believe it is, and the age range is Devonian to Pennsylvanian, you've probably misidentified it. So when you are analyzing and classifying D, it's a good idea to do the other four fossils first, because that's going to give you an idea about what kind of age range you should be seeing for D, and then that's going to help you with the identification process. Now, in the case of E, once again, we have another pill bug-like fossil. You can see that it has this smooth, looks almost circular head region. Then you have a middle portion that consists of just two obvious segments, and then you have a rear portion that actually looks surprisingly like the head portion. So the head and the rear are approximately the same size, and they're separated by this, middle region that just consists of two segments. Notice how that's different from A and C, where the head region is clearly noticeably larger than the rear. So in terms of this organism itself, the eyes aren't exactly that easy to see. However, you can clearly see in both the head and the rear regions, you have these noticeable raised areas, these lumps. And so the fact that the head and the rear portions are about the same size, the fact the middle portion consists of just two segments, and the fact that you have these raised portions in the head region and the rear portion, that's going to help you with your identification. And so hopefully you can then identify all 5 fossils. For location 1, you'll put their ranges within the correct columns. You will also name the species using the name within the fossil plate, and you will write it in here. And then hopefully once you've put all the ranges together, you will see that there's only one period in which all of these fossils could have coexisted. So you'll be looking for a region, let's say the Silurian, and so all of the 5 fossils will have been present in the Silurian, and that will have been the only time when these 5 fossils could have coexisted, and therefore you know the age of this rock is Silurian. Okay, so now let's have a quick look at the fossils for location 2. Okay, so once again, at location 2, we are straight in with fossil A, and straight away you can see we have another pill bug-like fossil, don't we? So you can see we have a head region, we have a segmented middle, and we have a non-segmented smooth rear portion. You will notice that there are clearly eyes. You can see one eye here, and you can just make out another eye here. You will notice that that nose region that we saw on some of the other similar looking species is not present in this instance. So the head is actually pretty smooth. And the same goes for the rear portion, which is also very, very smooth. And so this is a rather distinctive looking fossil because when you look at the head of it, it doesn't have this kind of semicircular shape that you're seeing with examples like this or this. Instead, the head is, it's rather distinct in its appearance, to be honest. So you should be able to identify this particular fossil relatively easily. Now, B, when you look at it, B looks like the kind of sea snail fossil. So it looks like a gastropod. I'm pointing out right now, I'm saying it looks like a gastropod. I'm not saying it is a gastropod. So what can you see? Well, you can see that the fossil itself clearly has a pointed appearance. It has this cone-like appearance, and you can see that it has separate segments which are creating a spiral-like pattern on the shell. Now, the next thing you'll notice is the shell itself is actually quite smooth. There are no obvious lines on the surface of the shell. So there are no lines coming from the tip towards the bottom here, or there are no lines which are running parallel to the spiral of the shell. So you have a relatively smooth shell which has this distinctive spiral pattern to it. And you can see that the shell itself has quite a distinct point to it. So it's closing up into quite a tight point. It's not a very broad, relatively flat spiral. It's quite a tight spiral that's forming this very obvious pointy triangular shape. Now, once again, just like fossil D from location 1, this is a morphology which is surprisingly common. So you'll see a few examples of shell shapes like this within the fossil table. So straight away, you're probably thinking, how do I identify this fossil? So in this instance, what you're going to do is you're probably going to wait to identify fossil B, and you're going to analyze the other fossils first, get the age ranges for those, and then you're going to look through the fossil, the possible fossils, which look similar to B, and you're going to say, right, which one of these fossils seems to fit in with the age range that the other fossils are suggesting. So in the case of fossil C, we're into this brachiopod bivalve situation again. You can see it's clearly a clam-like shell. Now, in this case, you will see that it does have a very distinctive shape. So this is the hinge back here. So obviously where the two parts of the shell actually hinge on one another. And this is the margin here, which closes up to actually seal the animal inside the shell. Now, straight away, you will notice that the rear of this shell is very, very flat. It's almost a straight line. So that's a bit of a giveaway straight away about the morphology of this shell. You'll also notice that it does have a rather distinctive shape to it with this flat back and this sweep coming around here. You will also have spotted that you have abundant striations emanating from the hinge region and moving out towards the margin in this sunburst-like pattern. So the fact that you have this rather flat hinge region and these radiating striations is going to help you identify this fossil relatively quickly. Now, In the case of D, once again, we're in this brachiopod bivalve situation. The first thing you've probably noticed is that the striations on this particular shell are very, very deep and they're quite wide. So compare that to the striations for C, where you'll notice that they're quite thin, so that you can see them, but you can't really pick out individual striations very easily. Now compare that to D, which I'm just going to zoom in on slightly. You will notice straight away that these striations are a lot more visible. They're a lot wider, they're a lot deeper. So when you are looking at the fossils in the fossil tables, you will be looking for a diagram that clearly has very broad, very deep striations on the shell. Now, when you look at the hinge region for the shell, you will notice you have this hinge region right here, and then you have these, these flared regions which are coming out here and here. You should be keeping a lookout for those in the fossil table. Now, another thing you'll notice is when we're looking at this particular fossil head on, so front on, you will see that it appears to have a region which is a trough. So there is a depression in the shell. So the shell's coming in like this, then you have this depression, then it comes back out on the other side. So it looks like there is some kind of depression in the shell. You can see it in this front-on view, and you can also see it in the view from the rear. You can see there's that depression again. So you're going to be looking for a fossil that has this feature. And the final thing you're going to notice is that where the two halves of the fossil meet, where the two valves meet, you will see that it has a zigzag, a sawtooth pattern. Now compare that to fossil C, where the edge is relatively smooth. So where the two halves of the shell would meet, you would have a single line, and it would be flat. In the case of D, you can see that where the two valves are meeting, you have this rather distinctive zigzag sawtooth pattern. So once again, you're going to be looking out for that, because that's going to help you identify exactly what species you are looking at. Now, if we move down to E, what can you see? So in the case of E, you can see you have, quite a complicated looking fossil. There's a lot of things happening there, but they're all quite simplistic. So straight away, you can probably see you have what looks like a broadly radiating pattern. So you can see you have lots and lots of lines that are coming up from this core region here towards the edge of the fossil. And you can see, you can pick them out. Here's one right here. You can pick out another one here, another one here, and another one here. Now, you can also see that you clearly have lines running perpendicular to them. So if we look at this line here and this line here, you'll notice that you have a perpendicular line here, 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 and here. So clearly, this particular fossil consists of chambers. And the question is, well, what shape are these chambers? Well, in this case, they look approximately square, but you can also see in the case of this region here, they appear to be rectangular. So clearly what you're looking for is you're looking for a fossil that has multiple square and or rectangular chambers. It looks like it could have a radiating pattern, so keep a lookout for that. And you're going to look for something that kind of looks a bit like a wire mesh. So you're going to have these vertical portions here, and you're going to have horizontal portions as well. So once again, if you're having difficulty identifying fossil E, it's a good idea to identify the easier fossils first, because that's going to give you an idea of what age range you are looking for. And so that might aid you when it comes to identifying fossil E. And then finally, for fossil F, well, fossil F has this tree-like appearance. So you'll see you quite clearly have this stem region here. Then you have this region, which I'm going to simply refer to as the bulb. And then coming off the bulb, you have multiple, well, how can you put them? Branches. And you can see that these branches actually end in very fine feather-like features. Okay? You can see them when you zoom in. You can see several of them here. You can see some here, and you can see some up here as well. So this shape is quite reminiscent of a particular group of organisms. You should be able to find it in the fossil plates relatively easily. the question then becomes, well, which of these species for this particular group of organisms am I looking at? So let's look at some of the features. When you look at this bulb region here, you will see that it consists of several approximately equal but quite irregular looking plates. So it looks like it consists of several plates rather than one plate by itself. It looks like you have multiple plates covering this bulb region for this particular fossil. You will also notice when you look at these branches, you can see that they clearly consist of individual segments stacked one on top of another. So you're going to be looking out for features like that. You'll also notice that this particular branch here, it bifurcates. So it starts down here, it comes up, then you have one part goes up here, and you have another part coming up here. So you're going to be looking out for that as well. Some examples of this particular group of organisms, the branch won't bifurcate. It will just be a single branch. But in this particular instance, you can see that they will bifurcate. And then you could also maybe, when you look at the images in the fossil tables, you may also be able to see in those images that you might have these finer feather-like features in the diagram as well. I'm going to be honest, for this particular species, this particular organism, I can't remember whether these finer features are located in the fossil diagram, but if they are there, that might help you with the identification of this particular organism. And so once again, you are going to put the ranges for each of these organisms into your diagram. Here, for location 2, you are going to write the name of each species as it's given in the fossil tables in this area, and then you're going to date the lithology. Once again, there's just going to be one period where all 6 of these fossils could have coexisted, and so that's going to be the age for your lithology. Now, once you've done that, obviously you're going to be putting the ages into this table here, but please don't forget you are also going to be writing them here as well. All right. So once you have dated each of the layers, you are going to put them into that table there. Now, once you have done that, we are then going to move on to question 2. And we are going to deal with question 2 and question 3 in the next video. So if you want to stop, get up, have a walk around, take a few minutes, maybe get yourself a glass of water, and I will see you in part 2.